Hello friends, today we will discuss about blue-white screening, a method which is widely used in recombinant DNA technology for the identification of recombinant bacteria. So what do you mean by recombinant bacteria? Recombinant bacteria are basically bacteria which are carrying our gene of interest. Basically they contain a foreign plasmid which contains our gene of interest where to express our desired protein, which is widely used for industrial purposes and many other purposes. So we can see a vector which is widely used, having a ampicillin marker, a lag Z marker, and a ori region. The ampicillin marker in this case is widely used to help us to identify the transformed bacteria. Suppose any bacteria which carries this foreign plasmid or any bacteria which has been successfully transformed will be carrying this plasmid. Now this plasmid has ampicillin resistance gene, so we will be getting a protein which will degrade ampicillin. So for those bacteria which will be carrying this plasmid, they will be able to degrade ampicillin if the media contains ampicillin, so they will be able to grow because ampicillin is a antibiotic which resists the growth of bacteria. Again, the plasmid also contains lag Z marker. Now this lag Z marker basically codes for lag alpha subunit of beta galactosidase. The process is also known as alpha complementation because in alpha complementation the alpha part is produced from the foreign plasmid. The alpha subunit of beta galactosidase combines with the omega subunit which is coded by the host chromosome. The alpha and omega subunits combine with each other to form a functional beta galactosidase which will basically cleaves your galactose. Okay, and it also contains ori to allow its independent own replication. So what happens in case of blue-white screening? Let's see. So this is a foreign plasmid. The foreign plasmid contains your ampicillin resistance gene, the lag Z region, and the ori. The lag Z region contains MCS, multiple conic sites. So what do you mean by this MCS? It means this site contains the lag Z gene basically contains a site known as MCS site which contains multiple restriction enzyme recognition site. That means if we cleave with a restriction enzyme whose recognition site is present within this MCS or multiple cloning site, this plasmid will become linearized. Okay, this plasmid will become linearized. Now we will take our foreign DNA which we want to insert into this plasmid and which we want our bacteria to express. So we will take up foreign DNA which has been treated with same restriction enzyme so that the ints generated here and the int generated in this plasmid will be complementary to each other so that they can bind with each other. Okay, so we will take plasmid, our foreign DNA, we will cleave it with the restriction enzyme, we will mix them, and in many cases we will see the foreign DNA has been because of the complementary ints, they binds with each other and foreign gene gets inserted within the plasmid. While in many cases we will see the plasmid itself get recircularizes without the insert of foreign DNA. Now both the plasmid has ampicillin resistance gene. Okay, but the difference between them is that this plasmid is a recombinant one because it contains our foreign DNA, while this plasmid does not contain our foreign DNA. Okay, so but we don't know that which one is recombinant or which one is non-recombinant. So we will transform our bacterial cell with those plasmid. The bacteria will take up using transformation process. Transformation is a process by which bacteria takes up your foreign DNA. So the method used widely, electroporation calcium chloride method. So okay, so when this foreign plasmid gets inserted within the bacteria, and the bacteria itself starts to grow in this ampicillin plate, so we will get blue colonies and white colonies. Now why do we get these blue and white colonies? Let us see here. So I have said you that the lag Z region of the plasmid contains basically your insert MCS site. The MCS site within this lag Z allow the insert of foreign DNA, because we will cleave the restriction enzyme. The restriction enzyme will recognize the restriction site present in the MCS region. Will recognize the restriction site present in this MCS. MCS is nothing but the collection of many different restriction site. Okay, so restriction enzyme will cleave the restriction site, and the foreign DNA will be inserted. In many cases, the way it itself get ligate to each other, a functional alpha subunit will be produced. Okay, now this alpha subunit will combine with the host omega subunit to form a functional beta galactosidase, which will cleave this chromogenic X scale substrate to produce blue color, and those colonies will be giving blue colonies because their laxate region do not contain any foreign insert. Okay, so there we will be getting a functional beta galactosidase enzyme. While in those cases where foreign gene has been inserted, laxate frame has been distorted, so no functional alpha subunit is produced. So here we will get non-functional beta galactosidase, and XCal will not be cleaved. So in this media containing ampicillin plate, again remind, I have said you that ampicillin is present in the vector to allow only those bacteria which will take up this plasmid, because bacteria do not have itself the ability to grow unless and until it is a drug resistance one. But normally we use lab strain, so it will only allow those bacteria to grow in this amp plate where your foreign ampicillin resistance gene is present in the foreign plasmid, and it will cleave the ampicillin within the plate and it will grow. But in one case the recombinant plasmid is present, and in other case the non-recombinant plasmid is present. So non-recombinant plasmid will be able to grow, but they will be giving blue colonies, and those recombinant ones will give us the white colonies. So this is how we get to understand about blue and white screening. Hope you have understand the video. Please click like and subscribe to my channel. Thank you.